Start recording. So good afternoon, folks. Thank you for tuning in to another Risk Institute online session. Today, we're fortunate to have with us the wonderful Miss Peg Coleman, who will talk to us about her research on incorporating food and gut microbiota in the 21st century risk analysis. The presentation will be around 45 minutes and with around 20 minutes of Q&A. Please could you make sure your mics are muted, and if you'd like to leave any questions in the chat, Miss Peg Coleman has expressed her desire to have an open conversation, so she doesn't mind if we interrupt her with questions during her presentation. But before we begin, I'd like to make a few announcements. So our next talk on the 25th of January around 1 p.m. Is Professor Ekaterina Auer. She will talk about towards assessing the likelihood of mutations in the BRCA1 and 2 genes with interval and Dempster-Schafer theory-based methods. Then we also have the Risk Institute annual showcase on the 1st of February at 10 a.m. Dominic Hose on the 15th of February. And we also have Professor Zafalon Marco on causal invariance in precise probability in action on the 22nd of February at 2 p.m. So I'd like to introduce our speaker, Ms. Peg Coleman, who is joining us from New York today, is a medical microbiologist and microbial risk assessor, a fellow of the Society for Risk Analysis. Ms. Coleman has served in many leadership roles in the SRA since 1995 and was recently elected to be on the SRA Council. She presented a series of SRA webinars in 2017 and in 2021 on topics related to the microbiome revolution. So Ms. Coleman has served as a mentor for graduate students and new members in the SRA mentoring project and has a long career as a microbial risk assessor, began with the US federal government and continues as a consultant. Primary interests are benefit, risk analysis, and the resilience of the human organism. So she's got a breadth of experience into areas of well-being expert, publishing 55 technical reports and peer-reviewed manuscripts on microbial risk assessment, and shares into transdisciplinary collaborations in Cornell University and with a professor in immunotoxicology, and has a series of manuscripts on the microbiota of milks and resilience of the human superorganism in health and disease. So she provided a lot of inspiration for our raw milk series. So please let's welcome Ms. Peg Coleman, and if you'd like to start sharing your slides.
Okay. Oh,
let me stop sharing here, sorry. Yeah. There you go.
All right. Well, good morning. I'm really delighted to be here. I'll get right into it since I have quite a few slides to discuss with you. This is a brief outline. I always start with introducing the human superorganism and how I came to understand it and the need to incorporate it into risk analysis. Then a surprise for me, how much interaction there is between the gut-lung axis, the respiratory system, the brain in health and disease. Then a few slides on managing our microbes for health protection from disease. Some evidence maps on benefit risk analysis for mammalian milks. And then I'm hoping to hear some dialogue about how we might incorporate the food and gut microbiota into 21st century risk analysis. So here we go on human superorganisms. I had the pleasure of meeting Rodney Dietrich at a book signing. And I didn't know at the time that he lived half an hour away from me and worked at Cornell University. So when we met in 2016 around this book, The Human Superorganism, that started a whole series of collaborations that have really transformed my thinking. So this idea, and excuse me, I have to move my tab so I can see the slides better. Does that interfere with you when I'm moving the tab, the Zoom tab?
Not at all. Not at all. It's stayed the same the whole time.
Okay. So current knowledge in the 21st century, sterility is not healthful and may actually be harmful. So the idea that Rodney put out in this 2016 book and his webinar in 2017 on protecting the human superorganism is that Homo sapiens is incomplete without its microbiota. So together, the human genes and the microbial genes form a holobiont or a superorganism that needs to be considered when we're thinking about risk. And the new medical landscape that's emerging has been even more accelerated with the challenges of COVID, and how might we manage our microbes in a different way than just relying on vaccines. Rodney previously had published this paper with a colleague from Johns Hopkins on inserting the microbiome into our frameworks for risk assessment, that really exposure doesn't go right to the host cells. If we are talking about exposure to a chemical or a microbial pathogen, it's the microbiome that is the receptor before we even get to an internal dose in a host cell. And so there's a need to expand our frameworks for considering how the microbiome relate in the human superorganism. This is a depiction of the traditional framework for microbial risk assessment. It's probably familiar to you all, but hazard identification, exposure assessment, dose response assessment, risk characterization. What may be different for you is considering threshold and non-threshold models for dose response assessment. That is one of the keys for transforming our thinking in the 21st century to account for the protections of a healthy microbiota, and to not assume that one of any microbial pathogen can infect and cause illness and disease and fatalities. The motivation for some of our work with Rodney and others was this paper by Wiedemann in 2011 on evidence maps. And this was appealing to us because we can accommodate more and different types of information. Also, there's an openness and a transparency to this process. It provides a graphical representation of the evidence basis, and you'll see this later in the presentation. And it's structured as pro and contra arguments with supporting and attenuating data. So we find this, this framework more responsive to the different sources of information from multiple disciplines that we're called to look at more holistically. And also this evidence map framework assists risk analysts in avoiding traps like confirmation bias. So seeing in a study what you already believed before you see the study. And the evidence map really helps people to step back from their beliefs and opinions and look more objectively at a body of evidence that may be surprising. I'm hopeful that the evidence map facilitates constructive dialogue, and we'll roll around to that again at the end of the lecture. And especially when we have diverse experts and non-experts that all want to understand the evidence, we're hopeful that this is a framework that's helpful in the future. It was applied in 2011 to nano materials, which was quite controversial in its day and maybe is less so now. But I would say that the microbial phenomenon, it may be helpful since that is controversial in some areas, especially around raw milk. So this is a template that we used in one of our papers. We have a series of 3 papers in applied microbiology with Roddy Dieter, with D. Warner North, and another colleague, Michelle Stevenson. So this template was presented in an evidence or enhancing human superorganism ecosystem resilience by holistically managing our microbes. So it sets up the evidence basis, conclusions, remaining uncertainties in one column of the evidence map. Then you get pro and contra arguments in the middle column and mechanistic studies on the left. So it's a synthesis of a lot of information and more technical information than any normal person would want to hold in their mind for one paper. So we ended up parsing different pieces of our work on evidence maps into 3 papers to really be able to develop it in a way that we're hopeful assists in deliberating the evidence. So now we'll jump into section 2 on health and disease. And I was a regulator in the 1990s, and these symbols were developed in the US, and it reflected a common worldview that eliminating bacteria in foods was our goal. And so this fight back, this microbial monster that was used in communications of risk, that's slightly changing over time, but since 2015 in the US, and I would expect in the UK this is true too, that there's monitoring by whole genome sequencing. And so there's much richer information available now on the variability and virulence profiles for human foodborne pathogens. And in the past, microbial risk assessors might assume that the only representatives of microbial pathogens were the highly virulent strains that caused outbreaks. And so we do have an expanding window of knowledge to incorporate. Also, this, there's one more figure, okay. There is a recognition on the part of regulators around the world that maintaining food temperatures limits the pathogen growth. And so 40 Fahrenheit or 4.4 C, this little figure now has fight back being frozen, but still a threat. And I draw your attention to a European Food Standards Authority paper, a scientific opinion on public health risks related to the consumption of raw drinking milks. That's an interesting study, and they do mention control, temperature control being effective in managing risk. So a typical 20th century assumption about superorganisms, neonates, infants, and young children are highly susceptible to all infectious diseases. I think that might still be a common assumption, but it's actually untrue for some pathogens. And C. difficile or Clostridium difficile, abbreviated CDI in a few of these slides, it turns out that neonates and infants are resistant, that there's a high rate of asymptomatic colonization, very rare symptomatic CDI, but that it disproportionately and severely affects the elderly in nursing home environments and also in hospital environments. It's true for Staphylococcus aureus and hospitalized neonates. We actually discuss C. difficile and Staph aureus in our Managing Our Microbes paper that you'll see referenced at the end of the talk. And I point to one future growth area that pathogen microbiota host environment specific dose response models might be needed. There certainly is so much variability in human superorganisms that just taking one data set and extrapolating to all these different populations may not be helpful in managing the risks. So the primary risk factors for hospital-acquired diarrhea, and I would say some other hospital-acquired infections, CDI, exposure to antibiotics, polypharmacy, chemotherapy. And these are important because they knock down our innate protection of our healthy gut microbiota and make us more susceptible. The duration of hospitalization influences risk, age, and underlying comorbidities. We've seen this list in COVID-related papers for some time now, so that's probably second nature for you to realize all the different factors that are related to risk. Vitamin D deficiency, for some reason, is important for hospital-acquired diarrhea and CDI, and GI surgeries. All of these, well, many of the slides, I list one reference at the bottom, and I can certainly give you more information if you're trying to track that paper down and you can't find it. There are interconnections between diet and dysbiosis that contribute to physiological declines with aging. And this particular study I found really helpful to think about dysbiosis and malnutrition, which of course is a global problem. And then the gut-lung axis, all those contribute to age-related respiratory diseases. So cognitive, sensory, these are neurological connections to the gut microbiota, immunity, and even musculoskeletal effects can be related to inadequate diet or malnutrition. So that's an important factor to look at, especially as we're trying to figure out how to find a new normal for populations in as the pandemic continues. Here's an older paper, but a very helpful one in thinking about a term that's just starting to appear in the risk literature, colonization resistance. So it turns out that there are related commensals to CDI, to C. difficile, that influence or protect against its growth and infection in humans. So there are direct mechanisms, indirect mechanisms, but the focus here, this is a related Clostridium strain that has been associated with protection from C. difficile. And part of what's the knowledge that's evolving is that short chain fatty acids, or SCFAs, are an indirect mechanism that promotes colonization resistance to CDI. And we cite a number of papers that will help you dig deeper into the literature about colonization resistance, but it's essentially innate resistance, very different from the vaccination strategies that we think of for COVID. Oh, and here's my text box telling you what this SCFA is, short chain fatty acids. So butyrate, propionate, or acids like that. And I wanted to give you just one more slide on the related commensals. So here's this commensal strain, and under conditions with a healthy microbiota and no antibiotics, there are interactions with C. difficile that limit its growth. But if you give a broad spectrum antibiotic and you knock down the healthy gut flora, you create an opening, you inhibit this related commensal, and you allow a niche for C. difficile to affect. You remove the block. And part of the hospital-acquired illnesses is looking more at the ecology and less at treating everything with treating with antibiotics or with sanitizers. Sanitization might not be as helpful as we think. So I've been interested in the gut-lung axis, but it wasn't until this last couple of years that I appreciated the connections. So most people would realize that we have a gut microbiota and that you can have from 100 to 10 to the 12th organisms in our GI system in different parts. So there are gradients of organisms, associations of organisms, but there are also microbes in our lungs, and not to as high a degree, from 100 to maybe 10 to the 5th per gram of tissue. And our oral and nasal cavities also have microbiota associated. So you have colonization in your nose and mouth. You can have interconnections with mucociliary clearance, with gut reflux. And then there are mechanisms proposed for circulation by the blood and the lymph of the microbes, of their metabolites or metabolomics, and immune cells. So it's really fascinating the kinds of interconnections that we're discovering, that the gut microbiota affects so many other systems, the respiratory system and even neural systems. So for this slide, the gut microbiota status in COVID-19 is starting to get some attention. So 2020, this is one of the older papers now in the COVID literature there. But the idea is that for health, eubiosis versus COVID-19 infection and dysbiosis, there are connections that seem to be gut related. And I had just this fall heard the term long COVID, and we'll come back to that in another slide. So gut dysbiosis, influenza and pneumonia, this is another slide that helps us think about how these interactions might actually happen. How is it that the microbes in our gut are influencing our lungs? And again, this term SCFA, short chain fatty acids, those are the metabolites that seem to circulate and to be associated with either killing pathogens or promoting commensals. Oh, yeah, I did on most of these slides add a little box that reminds you what short chain fatty acids we're talking about. This is a paper that shows the lung microbiota landscape of COVID-19 patients. And I really, to be honest, I had not thought about having a lung viriome, but we have lung bacteriomes, so the bacteria in that are colonizing the lung, but there are also viruses there, and 18 of them in the healthy humans that were, well, let's see. I'm not sure if this was a comparison between healthy and control or just characterizing the microbiota of the COVID patients. I'll have to look at that paper again. But there are correlations that suggest that the lung microbiota merits further studies on mechanisms and causation to manage the microbes of the respiratory tract for COVID patients. This study introduced me to this idea of long COVID. So there are 7 healthy controls and 7 COVID cases. And we're shown the community abundance on the phylum level and on the genus level. And part of what seems to be observed in many systems of health and disease is that in health, you have a greater diversity of microbes, and somehow the individual taxa of the microbes that are present may be less important than the diversity of them. So that's certainly worth looking at more clearly. And when I searched this week on long COVID, I wanted to point you to a few studies that seem really interesting for risk assessors to think about. So here's a systematic review of persistent symptoms. These are all free full text open access papers. Here's another systematic review on fatigue. A follow-up of those with mild disease. So more often we see papers that describe cohorts of the more severe cases, the hospitalized cases, as suffering from long COVID, but there are some effects even for the mild cases. And this was an interesting title, The Kids Are Not All Right, that university students also report post-COVID syndrome that still influences their daily life. So we have a rich body of evidence on COVID and the symptoms not stopping when the infection stops. And what's that about? And how do we need to look more holistically at the human superorganism to help these people recover? So, oops, I went back. So now we're at section 3, managing our microbes for health and protecting against disease. This is a graphical abstract for our applied microbiology paper. And you will see the term colonization resistance here for a healthy outcome where you have a diet that has fresh whole foods, beneficial microbes, prebiotic factors, and you have a healthy human superorganism, not just microbes on your hand, but probably more important in your gut. And that those kinds of exposures are more likely to lead to well-primed immune systems balanced for inflammatory response to microbes in the environment. And I'll get back to this idea. Recommended daily allowance for vitamins. Is it time that we started thinking about a recommended daily allowance for microbes? Do we need more microbes in our diet to be able to maintain a diverse gut microbiota that's more resilient to challenges? in defining the dysbiotic human superorganism, we point to the diets that are increased in processed foods, lower in beneficial microbes, even the effects of pharmaceuticals, creating open niches for opportunistic pathogens. That's where you have dysbiosis, disruption of colonization resistance, imbalanced immune systems. So you can read more about this work and the examples that we give in this paper. In addition to C. diff and Staph aureus, our third example for this Managing Our Microbes paper, I mentioned to Francis, there's a link between autism and gut health. And so I point you to this paper if you'd like to read more about that. So the diet affects the whole superorganism, and this is a novel way of thinking, that there's a gut-lung-brain axis, and what is it that's transmitted between the gut-lung-brain axis? And it seems to be, again, these short-chain fatty acids, and they influence every system in our bodies. And it's fascinating to think that a healthy gut influences so much of the biology of our internal systems, many of which we had thought in the past were sterile and needed to be sterile. So this is really crucial, I think, to look mechanistically at what is communicated in health and how can we intervene in disease. So there's crosstalk between the gut microbiota, the immune system, and the lungs. Asthma is mentioned in this particular figure. Again, there's so much fascinating work on gut, lung, brain axis. If you need a PhD project, I think you could find a lot of material there to wrestle with. But again, short chain fatty acids, those are the communicators in the system. And the idea of managing our microbes for health and resilience, if we have poor quality diet, what are the consequences? We might have reduced microbial diversity in our gut. We might have pathogen infection, inflammation, erosion of the mucus layer, and intestinal permeability, so leaky gut. All these might be related to the diets and to low short-chain fatty acid levels. Now, for a healthy diet, that's associated with higher microbial diversity, with colonization resistance to pathogens, and with higher short-chain fatty acid levels. And I would add to the healthy diet whole dairy and animal and fish fats and proteins. So certainly the nutrition literature has changed as we've understood more about the microbiota, and we'll get back to whole dairy. So resilience of the healthy gut microbiota. If you're wondering how people think about resistance, it's the ability of the system or an ecosystem to sustain or restore its basic functionality following perturbation, challenge, or stressor. And a healthy gut ecosystem is just fantastically complex. There's high diversity and richness, over 2,000 species, 12 phyla, 9 dominant genera, and there's also functional redundancy that it might be a whole consortium of organisms that process nutrients sequentially and simultaneously that contribute to high richness and diversity. And the stressors for gut resilience include pharmaceuticals, and antibiotics are a big challenge, malnutrition, and diets low in fiber and enriched in processed foods. So these are some of the pharmaceutical stressors, antibiotics, even laxatives, NSAIDs, protein pump inhibitors, polypharmacy. They all can influence the gut microbiota. And I wanted to show you one slide of gut microbiota because it's just so dramatic when you look at the diversity of healthy controls and then the blooms of opportunistic pathogens for these children that are undergoing long-term IV nutrition. And IV nutrition just is not complete nutrition, and these children are suffering and they may not recover. But how do we manipulate a profoundly dysbiotic system for these severely ill children and restore the balance, the diversity and richness of the healthy gut microbiota? So we made it to the evidence maps, and you may know that mammalian milk is a 200 million year old superfood, and the emphasis has waxed and waned over centuries, but now, even though wet nursing was an ancient practice and it may still be practiced, excuse me 1 minute. He was running on my monitor. It's not very helpful. Okay. So the World Health Organization recommends exclusive breastfeeding for at least 6 months, the first 6 months. And we know that breastfeeding reduces the frequency and duration of respiratory and diarrheal illness in infants, and that exclusive breastfeeding protects against common infections and lessens the frequency and the severity of infectious episodes. So that's all pretty well accepted in the scientific community. But this recent study, also from University of Liverpool, I don't know if you all know George Okonomo, but this paper describes the milk microbiota, and he looked across human, cow, water buffalo, goat, and sheep, and he coded the organisms that were present in every one of these animals in red, in yellow at least 3, and in blue it may just be 1 or 2 where there are common representatives in that genus across the host species. So there are similarities and there are differences. These are the human microbiota studies, a Venn diagram showing for 4 studies, the similarities and differences, the shared species in the middle in the red. And here are a few of the microbial species. And you'll notice similarities across the human bovine divide. And this figure is in the supplemental materials for our applied microbiology paper. And this is a graphical abstract from our donor breast milk paper. So similarly to the previous graphical abstract for the benefits of raw and pasteurized breast milk, diversity of gut microbiota increases, colonization again increases. There are other effects. You may not realize that if a mother can't breastfeed her child, she can go to a donor breast milk bank, but they pasteurize the donor milk. And I had not realized that until we had an SRA webinar in 2017. And that kind of started me exploring the milk microbiota in more detail. But if you pasteurize donor milk, you have obviously decreased diversity in the milk and in the gut microbiota of those infants. You have more dysbiosis, you have lower colonization resistance, and increased risk of other diseases. So our premise was, what are the benefits and risks of raw and pasteurized breast milk? Because the assumption that pasteurization increases benefits and decreases risks does not seem to be consistent with the evidence. So here's a general view of the human milk bank policies. There are countries that choose not to pasteurize donor milk, Germany, Japan, and Norway, for example. But the assumption that pasteurization minimizes risk for NICU infants may not be true. I won't go into these individual studies, but I want to point out that for this, the population of infants that is considered vulnerable, the choice of pasteurizing human donor milk is a more complex one. That the loss of benefits for pasteurized milk from these clinical studies all around the world suggests that we need to look more closely at the evidence and really deliberate our assumptions in light of the evidence. So here is our evidence map for breast milk, and I won't go into much of it. I point you to the paper, but each of these studies in the evidence basis that's listed here is described individually, a short synopsis. And so you get a sense of what the evidence basis is, what the conclusions are at this point, and what the remaining uncertainties are. And in an appendix, we give some information about these mechanistic studies that support the arguments. So that's all I'll say about the breast milk evidence map, but I'll definitely be happy if you read the paper and email me with questions about the evidence map. So this graphical abstract is from a comparable paper on cow's milk. And again, we're acknowledging that the benefit risk weighing between pasteurized and raw may not be what is assumed from 20th century science. That microbiota laden complete foods like raw milks benefit the human, the microimmunosome. So that may be a new term, and I point you to the paper for a little more, but not just reduced non-communicable disease risk, but also reductions in infectious disease risk. Whereas the risks may be lower from pathobionts than expected in the past. And similarly to the breast milk evidence map, I won't go into much detail about this, but we do describe a discrete basis of evidence. We go study by study through the pro and contra arguments, come up with conclusions, remaining uncertainties, and the mechanistic studies supporting. So I think what's different about the evidence map for the milks is that we're now able to hold side by side infectious and non-communicable diseases. And that wasn't an option with the old framework that we used to use for risk. So I did point out just a few of the key studies that really conflict with the opinion that risk is higher for raw milk. And this is one study from European cohorts where there are comparable protective effects with breast milk and raw cow milk against respiratory and other infections. There are positive effects for even controlling for breastfeeding for raw milk consumption. And no clear associations reported for diarrhea and milk consumption from either pasteurized or raw. So that's an interesting study to take a look at. And here's a 2019 study. So we have 11 children who have allergenicity to raw milk, to cow milk, and they did a proof of concept provocation pilot. So that was a double-blind provocation. They tested with pasteurized milk or shot milk or raw milk and noted the volume of milk before an allergic reaction was triggered. And for raw milk, none of the children reacted to the maximum tested of 50 mils of raw milk. But for shot milk, some of these kids were responding with only half a mil of pasteurized milk. So there certainly is a difference in the way that raw and pasteurized milk is processed for these children that have allergies. And that study certainly is worth considering in the body of evidence. Oop, yeah. For functionality of the gut microbiota in children, I just put in one slide that brought us back again to short chain fatty acids and intestinal failure. Underappreciated corollary that there's broad diverse functionality and redundancy for gut bacteria that produce these short chain fatty acids. And a strategy for managing our microbes might be to replace antibiotic administration with a daily dose of a symbiotic, a probiotic and a prebiotic, or a whole food that contains these organisms. So these are, oops, sorry, let me go back. So here's butyrate was one of the short chain fatty acids that was produced by so many different families. It's so it's not just one organism or a collection of organisms. There's a whole consortium of organisms that can contribute to gut diversity and richness of the microbial flora and health. So there are some studies also about fiber, that inulin ameliorates type 1 diabetes, and short chain fatty acids, big surprise, they're again important in describing the mechanism for benefit of the microbes. I also wanted to say, as I had mentioned to Francis previously, that I am a daily consumer of raw milk. And in the US, it's a bit different from the regulatory system in the UK, where you can go to a farm and go to a vending machine and get access to your raw milk. In the US, there are some states, the green states, that allow retail access So you can go to a supermarket in California and buy fresh, unprocessed raw milk right off the grocery store shelves. But I live in New York State, which is one of the blue states where I have to go to a licensed farm and pick up my milk. So this is my milk and my kefir and our wood stove and our country kitchen here in upstate New York. And I do love my raw milk and my raw milk kefir, and I find as a microbiologist more compelling reasons to consider benefits of daily consumption of raw than pasteurized. So now we've had a look into the evidence and the interconnections of our gut microbiome with other systems in the body, and how might we incorporate the microbiome into 21st century risk analysis. So this particular figure draws attention to the interconnections between risk communication, risk management, risk assessment, and research, and that where the dogmas, the assumptions, opinions, or perceptions about risk don't match up with the scientific evidence, that analytic deliberative process is warranted. So some kind of specific process that can facilitate dialogue about complex issues. This analytic deliberative process was described pretty well in this book, Understanding Risk. It's available free for download. And the idea of building in cycles where research, analysis, deliberation, and interpretation is open and can include stakeholders and experts. Certainly, it's a mystery to some stakeholders what goes into a risk assessment, data and assumptions, and what comes out, estimates of risk and uncertainty. And the idea of building analytical deliberative process more broadly for the raw milk issues has great appeal for me and my colleagues. We actually were working to update the glossary for risk analysts. So this paper in 2018 included the definitions of colonization resistance and superorganism. So I think that more people are considering risk a bit more holistically, or at least they're acknowledging that our simplifications may not be warranted. Oops. And this example shows a dose response curve. So most of the microbial dose response curves use the log scale for dose. So this is 10, this is 100, this is 1,000 organisms, and the probability of salmonellosis. This particular curve was from a human study, and we worked from a mouse study and extrapolated backwards to consider the human superorganisms where their microbiota are disturbed. So this is antibiotic administration, and then, oops, I jumped the gun. So for someone with a healthy microbiome, they might tolerate 10 to the 7th or more than 10 million salmonella bacteria before half of them became ill. But for antibiotic dysbiosis, 100 organisms could make half of the people ill. And the curves in between represent longer periods of recovery. So this is recovery 1 day after antibiotics and then challenge 2 days, 3 days, 4 days. So by 5 days, we're back to about normal after antibiotic administration. And again, this was extrapolated from mouse experiments, but starting from the human dose response curve on the right. So what does this tell us? That there's indirect evidence that there's a 10 to the 5th protection as the magnitude of the effects for colonization resistance, considering mouse and human data. So a healthy human can tolerate about 10 million bacteria, where someone whose gut microbiota is disrupted by antibiotics may only tolerate 100 before they get ill. So that kind of thinking hasn't worked into our microbial risk assessments in any systematic way. The Milk Microbiota project started for me in 2017. We had a webinar series through SRA. Rod Dietrich was one, and my colleague Dee Warner North and I closed out the series with preparing to deliberate the evidence on benefits and risks. So now we have 3 papers published with Rod that lays out the evidence in a peer-reviewed paper, in a series of peer-reviewed papers. And I'll just jump down to 2022. We're seeking partners for developing international workshops to deliberate this evidence and the knowledge gaps for benefits and risks of raw milk. And it's certainly possible that University of Liverpool could host a workshop. So we will see what happens. The evidence, what is the evidence? I'm asking that question. What is the evidence that pasteurizing human donor milk and cow milk is beneficial to health? And I think I've walked you through the reasoning that we should deliberate the evidence more carefully and not just assume that the pasteurizing human donor milk or cow milk automatically reduces risk and increases benefits, because it doesn't seem that's consistent with the evidence. These are the 2 studies that got me thinking about daily allowances for microbes. And they're actually from a research group in Cork University in Ireland. And so they might be interesting collaborators to bring into the University of Liverpool. But it certainly is true, I think, that whole foods contribute more than just nutrients for human cells. That we're talking about an ecosystem in our guts, and we need to pay more attention to the human superorganism aspect of how we look at health and risk. So managing our microbes for health and resistance. I think I won't expand on any of those points. Certainly, what you see in the media and social media, in newspapers, even in scientific journals, may be a combination of science and opinion and outdated dogma. And it's up to us as risk analysts to look closely and systematically at the evidence and I list all these papers. I can send you any that you have trouble finding, but there are also links to these 3 papers in Applied Microbiology. And I thought for a university crowd, this would be an interesting way to close out this lecture. Rodney Dieter, my colleague who's an immunotoxicologist, he's done some work on serendipity, and serendipity you could think of as a talent for making fortunate discoveries while searching for other things. And I found this dissertation from Duke Divinity School that just blew me away. And this person wrote a dissertation called Symbiotic Grace, Holobiont Theology in the Age of the Microbe. And so she lays out a case where she says the microbiome troubles modern anthropology. It troubles modern theology and philosophy. And I would say the microbiome also troubles modern risk analysis. And I haven't met this woman, but I can't wait to contact her and to learn more about how she brought theology into the holobiont sphere. And yeah, I just thought that was too fascinating, too serendipitous. So here I've given you my contact information. D. Warner North has been my collaborator in these last few papers, and also Rodney Dieter. So I will stop there for questions. And shall I stop sharing or leave the slides up in case there are questions?
Yeah, I would say leave them up in case
Okay.
anyone wants you to refer to anything.
All right.
Well, thank you so much, Ms. Coleman, for an amazing presentation. That was really incredible, inspiring, loads of practical insights. There's no from a personal perspective. I thought there was loads of avenues to look into further as well. So I'll open the floor for questions. Just to get the ball rolling, I'll start with one. I was curious about the kind of recommended daily allowance of microbes that you could take from that graphic that you showed earlier. Is there kind of, how would you go about measuring that? Maybe it's a slightly naive question. I know this isn't my specialty, but if I wanted to say get my microbiota and gut kind of tested, how could I even go about that? Could you go to the doctor and get that tested and see what you were deficient in and what you should be taking to try and kind of course correct that? Or is there a lot of guesswork?
Well, I think that at this point, though we can appreciate that diversity and richness impacts health, I don't think there's a consistent way to say, well, what is a healthy microbiome? Right. And so attention has focused too much, I think, on the lactobacillus organisms, because there are a minor portion of some raw milk and kefir and yogurt products. Not so much the kefir and yogurt, but those organisms that have gone through the clinical trials as for probiotic supplements, they may help, but it's not clear to me that that we can make blanket recommendations for what microbes to add. Colin Hill actually asked the question, does it even matter? Do we just need to give a diversity of microbes and not certain microbes? So yeah, he's raising an important issue, but without a methodology to operationalize it.
Yeah, I see that as kind kind of a main pathway, I guess, for the risk analysis to take place on this, is to have at least an understanding of what those baseline values are, or what a healthy control is, and how we can start to course correct towards a more safer or more resilient system there. But I'll open the floor. I know there's loads of other questions I'd like to ask as well, but does anyone else have any questions from the floor?
Yeah, I do have one. So you mentioned term long COVID, and as far as I understand, it means like when patient experiences long symptoms. And I was wondering, is this a consequence of some specific conditions of microbiome, or does it mean that microbiome is unhealthy?
I had actually dropped in 4 fairly recent papers. Oh, I know. Let's see. Sorry, it might have been a more efficient way to do this. Where? Oh, that was it. Okay. So these are 4 recent papers that that looked at patients with mild and with more severe COVID, and I think it might be that with the systematic reviews that are available, that we might be able to say more about cause and effect, but I'm not sure we have the knowledge right now to say, are they just correlated, that people are unhealthy and they get worse with COVID and they don't recover? Or is it that they're deficient in some organisms, and giving them a probiotic cocktail could speed up their recovery? I don't think there's an answer yet, but I think that people are considering we could be using microbes as therapeutics and not just the vaccines. And part of the reason that the microbes are important is that that gives our immune systems 2 different ways to process COVID. It's the colonization resistance and the effect on the immune system that the microbes can alter. And but I think you're correct that right now we don't have definitive data to be able to point to whether this is just correlative evidence, or whether there really is some healthful therapeutic in the future that might be developed from these studies. So I don't know, I guess is a short answer.
Yeah, maybe we can turn this question into discussion. So just to, you know, just I'm happy that I can hear your thoughts and your opinion on this question.
Thank you.
I would like to add to that slightly, or go on a slight kind of meander. I know that some people argue that there's obviously physical symptoms with the kind of long COVID, and there's some people that argue that there's also a psychosomatic element that occurs with it. And then I know there's a correlation between stress and how that affects the kind of the gut, and how there might be a kind of correlation between, like, say, IBS and high stressful scenarios. I'm just wondering whether the 2 might be linked in that kind of aspect as well, whether those worrying about catching long COVID or worried about COVID might also have more severe symptoms because they're stressed and they also have a weaker gut, don't know whether you could talk a little bit about maybe how stress influences the
gut. And I think that this particular study of university students, that they did try to pull out metrics of stress, of just, I mean, they called it academic stress. So there certainly are people investigating across the different systems, looking at neurological, immunological, gut, and respiratory system. Yeah, and just clearing the infection does not always restore health. I guess that's the reality.
Okay, thank you. Yeah, because I think you're absolutely right. It's such a complex and nuanced area where sometimes these things can't easily be measured, and I think not everything that can be counted things that can't be counted can sometimes be extremely important, and that's where the risk analysis becomes extremely hard to conduct. Does anyone else have any thoughts on this at all, or any other questions they'd like to ask?
Hi, thanks for the presentation. It's very inspiring. I will get some raw milk after this presentation. Yeah, but I have a simple question. How can the microbes in the milk arrive to our gut? I mean, it will go through our stomach. It is very acid environment. Would that acid environment kill the microbes?
There are microbes that can survive. I can't talk now. There are microbes that can survive going through the gut acids, and the nature of milk is also fatty, and so the fat often is the matrix that will protect microbes as they're passing through the gut. And some of the probiotics that are sold are encapsulated in a slow release form to try and minimize death of the microbes in the stomach. But these foods are so rich in microbes that some loss still delivers some benefit, that there are still some survivors that get into the gut. And once they're in the gut, they have a favorable environment for forming consortia and proliferating. So I think in the whole milk environment, there's less pass through and decline in the gut ecosystem than perhaps in a food that doesn't have the fat that's protective. And there are people who drink raw skim milk, but I think that the balance of the fat is important to the microbiology of the ecosystem. So I usually do not choose to lose the fat.
Okay, thank you.
Thank you. I think we have time for 2 more questions. Does anyone have any more questions from the audience? If not, I'd like to ask about the evidence map, because I thought that was really intriguing. It kind of felt like a mini poster, almost in a more digested format, but a bit more comprehensive in a more systematic way. You said that they had quite a kind of, they were good for people that didn't have much understanding of the initial topic or didn't have any expertise in the area. I think because you went through it quite quickly, I didn't have a chance to look at it more closely, but I would love to look at this after the presentation a bit more too.
Mm-hmm. And I can certainly send you the links to the papers.
Please, yeah, I think that's a really nice way to capture a paper for sure in kind of a visual format. Did you present it to non-experts then and kind of measure their understanding or comprehension?
We haven't. In fact, we don't. my collaborators at present don't include a risk communicator, and so that's certainly important in the next step. When Mortar and I first started talking about the breast milk and bovine milk microbiota, it seemed that there were so many positions that were grounded in that 20th century science around the world. It's not just in the US, it's not just in the UK. And so we tried to pull together definitive evidence, and in these papers we give a short paragraph or two about each of the studies. And certainly the evidence is ambiguous in some ways. These studies are complicated and there are multiple ways to interpret them, but we tried to lay out what's known and what are the remaining uncertainties. And that to me is even different from the communications about raw milks that are commonly given by the regulators. Like CDC will still say that raw milk is an inherently dangerous food that they don't recommend for anyone. And yet they don't have an evidence map. They can't point to any studies that support that position. And so the idea of having a workshop and including stakeholders and regulators and consumers, yeah, it's challenging. And the consumers are not going to have the same level of openness to spending a day talking about these studies, even though us scientists, it's like, oh, that's heaven. But yeah, we haven't really figured out how to do this, but it might be that we need a series of workshops for different audiences. But certainly I would say this is way too much information for most consumers. We could not engage a consumer. Most
sure.
consumers.
Yeah, but this is really important work. We have a lot of convincing to do. If it's as important as all this evidence suggests, then there's so many people that I'm aware of that are scared of, in the public, that are kind of scared of germs and see them as a real kind of afraid of bacteria. You know, it's a phobia for many people. So
yeah, germophobia is real, and this is Rodney's book. I still keep it on my shelf, and
I need to check it out.
I definitely recommend this. This actually is accessible language for the public, even though he's an immunologist. This is much more common language presentation, and yeah, I think our education, and even from the fight back symbols.
I know, that was hilarious. I haven't seen it before, but yeah, that's so dated at this point.
Yes, it is pretty bizarre. But yeah, I do believe that microbes are our partners in health. And if we're regulating as though one of any pathogen is going to kill us, we're losing food that could be feeding the world. I mean, the idea of balancing malnutrition and food insecurity and all these other complications, it's not just about providing choice for those who want to drink raw milk. It's how do we help the developing world too, who have serious issues with their water and food and sanitation. I mean, it's there are no simple answers, I don't think, but
yeah, well, I mean, absolutely. I mean, even in the UK, there's many food deserts that we have, where in the middle of urban environments, there's people that don't have access to kind of raw and healthy foods, let alone something like raw milk, you know. Do we have any more questions from the audience? If not, I'll finish up on one last one.
Just one from me, if that's okay.
Please go ahead.
Hi, yeah, just wondering with the regulator's position, is it not driven by there's a risk of contamination and there's a sort of a low likelihood, high consequence downside risk to not pasteurizing milk, as opposed to the sort of high likelihood, but I guess still uncertain upside consequence of having the raw milk? So the regulators, you know, they're not going to get fired if they continue with recommending that we avoid raw milk, whereas if they stick their head above the parapet and suggest we don't need to pasteurize so much, then the danger is for them sort of personally in their careers, even though, you know, the evidence points increasingly in one direction, there's still that I guess worry for them.
So I think that your question is well framed, and I think that we are talking about an emotional fear of the infectious agents. And I didn't actually put a figure in the slide set, but our publication has a table that shows exposure studies from routine monitoring of raw milk in Canada, Finland, Germany, Poland, US, and even a UK study. And the bottom line is that across maybe 1,000 samples for each of 4 pathogens, the prevalence of pathogens is less than 1%, sometimes less than 0.1%. And so the risk, I think, is exaggerated by the emotional content of the decision makers, and not based on the evidence from well-managed farms. Yes, outbreaks can happen, but they happen in pasteurized milk too. And it seems to me that we can't continue to treat pasteurization as a silver bullet that increases benefits and decreases risks, because it's just not that simple. And there's evidence to the contrary. Yeah, so hello, he needs to say goodbye, I think. Yeah, it's not an easy fix, but and actually maybe the place to start is with the 2015 EFSA report. If you're curious about raw milk risks, take a look at that report. And I think they did a pretty good job of describing what's known, what are the remaining uncertainties, and how do we move forward. So yeah, I'm not really sure how to engage the regulators on this. That's going to take some negotiations, I think, to even get them to the table.
Yeah, I can imagine that. I think we'll wrap up, but I have one last quick question, if I may, Ms. Coleman. Sure. When you were talking about kind of the gut being affected and impacted by antibiotics, do you have any information or idea of how long it takes their gut to recover from that sort of situation?
There are studies that show return to normal within days, maybe within a week, but there are also people who suffer longer term effects. And I think because there's so much variability between humans, and it's the idea of defining a core microbiota has just not been a successful strategy. So what's healthy may depend on what other organisms are in your consortium.
Yeah, the definition of normal is already quite ambiguous, I guess.
Yeah. Or variable that there are. I've seen some. Actually, I showed one network diagram, but there are some network diagrams where it's once you have hundreds or thousands of species and think about all the potential interactions pairwise, and then groups of 6, groups of 10. I mean, it's an ecological I don't know if it's nightmare or oops, I didn't mean to do that. Yeah, the complexity we've only just begun to deal with in the gut ecosystem.
Yeah, well, that's ultra fascinating to me. So thank you so much for your talk.
You're welcome.
Yeah, no, I'd like to thank you on behalf of all the attendees and obviously the Risk Institute. It's been an absolute pleasure to have you on board. I look forward to passing this conversation, this presentation on to those that couldn't make it as well, and hope to see you again in the future.
Okay, thank you all.
Thank you very much. Have a lovely day.
Thank you.
Bye.
Thank you.
Bye.